an associate professor in the Department of Microbiology. I'm going to make a big shift to a lot more basic science. Frank asked me to talk about microbiomes, specifically in soil. So I'm going to give you a model example of what we've been studying in soils and how it relates to agriculture. So to start things off, just a basic definition. When we talk about the microbiome, what exactly do we mean by the microbiome? So essentially, you can think of the microbiome as all of the collection of living organisms inside any ecosystem, as well as all of the genetic information. So it's essentially the potential to do things in an ecosystem as carried out by living organisms. And so we've been studying the microbiome for hundreds of years, but only recently have we gained the tools to really answer some of the basic questions that we couldn't before. And so we've transitioned from being able to look at things under a microscope, then culturing things, only to realize that we could only culture a very small fraction of what's out there. And recently we've shifted to nucleic acid-based technology, as Gemma covered previously, with DNA and RNA sequencing that gives us access to everything independent of whether we can grow it or not. And the basic take-home has been that the more we look into what's out there, the more we discover. And so basically the complexity of the microbial world is drastically bigger than we had originally expected. And so if we think about these metagenomic approaches, what exactly are we getting from that? So you're sequencing all of the information in an ecosystem, and you're essentially taking a snapshot of what's happening in one time point. And it gives us an idea of what's there. It's essentially a census of organisms and their functions at any given time point in any given ecosystem. And so what we can get from that are quantifications of things like who's there and what are the functions that are there. And so this reflects the activities that are happening in any given ecosystem. And so what have we learned about New Zealand microbiomes, specifically focusing on soils? And so if I were to summarize it, basically it's that microbiomes are intricately linked to soil formation and management practices. If we understand how soils are made, we understand what microbes are there, and we can then modify them by how we manage our soils. And so if we think about soil as an ecosystem, we need to look at it from a microbial perspective. So the basic unit of soil is a soil aggregate. And the soil aggregate is an entire world. It's a whole planet for a microbe. And within that planet, there's going to be lots of different ecosystems. And so the complexity of the ecosystem is going to be reflected by the complexity of the microbiome. So if you have a very diverse microbiome, it suggests that you have a lot of ecosystems for different types of microbes. And so in that sense, microbiomes become indicators of what's happening in soils without having to measure absolutely everything that's happening in soils. So if we think back to how exactly did we get to how we are today in terms of microbiomes, we need to go back to the genesis of soils. And that goes back to things like geology and pedology. And so we have to look at the rocks. Basically, rocks are the foundation for soils, and they drive the evolution of different types of soils. And so if we look at the distribution of rocks throughout New Zealand, we see that we have a very strong influence of the mountain ranges throughout New Zealand that radiate different types of rocks as you go from the central Alps all the way out. And so this drives the formation of soils. And if we look at a map of the different types of soils in New Zealand, we see that it closely mimics what's happening. So we see a radiation of different types of soil coming from the mountain ranges out into the coast. And so we would predict, obviously, that the microbiomes should follow the formation of these soils because they're part of that soil. The problem with understanding patterns in microbiomes is that there's lots of confounding effects. And in New Zealand, one of the major confounding effects in understanding soil microbiomes is land use. So we know that 2/3 of New Zealand has been deforested and converted to grasslands. And so that overlays pretty well with the type of soils that we have. So we don't know at this point which one of the two came first. Did the land use drive the microbiomes, or did the soil formation drive the microbiomes and the land use? And so we need to be careful when we understand or we interpret patterns for microbiomes, because we don't really know which one was the driver and which one is the passenger. And so what we do is collect samples throughout New Zealand and try to look at these patterns and predict what are the processes that lead to the formation of soils and eventually the formation of microbiomes. And what we've learned both locally and internationally is that things like pH can play a really large role. And so this is a study that we did with an early PhD student where we collected soils throughout New Zealand. And we tried to account for things like the region that we were collecting from, the type of soils that we were collecting, and also the land use in terms of dairy, high country, or low country farms. And what we found was that if we look at all of the samples and compare them to the microbiome, we see a big role for pH in shaping these soils. And pH is directly correlated to the type of soil that we're studying. So all of these things are sort of touching on each other. But again, it's very difficult to quantify how important is pH for controlling these microbiomes and how much do they respond if we were to manage that. But it gives us an idea of what's happening in these soils. And so one of the secondary questions that we started asking is, what are the controls for microbiomes? How can we shift these microbiomes, and what are the repercussions? And so one of the things that we tried to focus on was greenhouse gas emissions, because we knew that that was a big important topic, and we wanted to use it as an ecosystem level process to look at the correlation between nutrient cycling and microbiomes. And we focus on pasture soils because they're the dominant landscape in New Zealand. So why did we focus on greenhouse gas emissions? We've had a few conversations here throughout the day. But essentially, it's because New Zealand is unique. New Zealand is very different from the rest of the world. So for instance, from 1990 to 2012, we were one of the few countries where emissions were still actually increasing rather than stabilizing or decreasing. This is for developed nations. Keep that in mind. And again, when we look at our split of greenhouse gas emissions, unlike the rest of the world where energy is the dominant source of emissions, New Zealand's greenhouse gas budget has 48% of our emissions actually coming directly from agriculture. So it means that we're different from the rest of the world, and so we need New Zealand solutions for New Zealand problems. And if we look at the split of emissions, we see that most of it are coming from dairy farming, followed by sheep farming and others. So it's linked to ruminants and how we actually farm here in New Zealand. But what I'm going to focus on is nitrous oxide, the secondary greenhouse gas that's associated with nitrogen deposition. So again, why is New Zealand different and why are we focusing on these emissions? Well, it's because our landscape is different and our farming methods are different from the rest of the world. So essentially, we have a very large landmass that's almost fully dedicated to grasslands. And in those grasslands, we're growing very large amounts of biomass. And that biomass is what's driving these emissions. So just to do a back of the envelope sort of calculation, so every animal that we're growing on a daily basis is doing this. And every time that they do that, they're depositing nitrogen into the soils. And that nitrogen is the source of the greenhouse gases. It's a very small proportion of the nitrogen that's deposited, but because it happens every day over very large landscapes with many animals, it eventually adds up to a very large proportion of our greenhouse gas budget. And so just to give you some context, so if we look at a single urination from a dairy cow, we can load nitrogen in a urine patch that's equivalent to fertilizing a paddock to the tune of 600 to 1,000 kgs of nitrogen per hectare. So really high nitrogen loading in a very small space. Now, if we multiply that by the fact that it's happening every day over multiple times a day in an area that's roughly 8 million hectares, and we're housing 42 million animals, we can see how the numbers stack up really quickly. So it's not a problem with the fact that the process is happening, is that we have so many animals and so much land that by adding all of those emissions, it becomes a very big number. But one of the things that it allows us to do is look at the nexus between soils, biology, and nutrient cycling and see how we can find weaknesses that we can tweak to our advantage. And what I'm going to show you is an example of tight linkages between soil biology, soil itself, and the biogeochemical cycles that control the flow of nutrients. And so one of the things that I'm going to focus on is rain, microbiomes, and emissions. So we got to remember that this is a biological system that has evolved over millions or not billions of years, and that there are legacy effects that we are ignoring potentially when we're interpreting data. And one of those is rain. So this is a generic plot for microbiome analyses. Each dot represents a sample from a different soil throughout New Zealand. There's 50 sites in this study throughout all of New Zealand. It's representative of all of the major soil classes. And so the dots are samples, and the distance between the dots represent the similarity between the microbiomes. Dots that are very close together show very similar microbiomes. Ones that are distant are dissimilar to each other. And so we can see that there's a gradient of microbiomes, and we've color-coded it based on rainfall. And we see that the distribution of microbiomes is directly and strongly correlated to the amount of rainfall. So rainfall is a historical driver of evolution for these microbial communities. Now, we took 20 of those samples and took them overseas and did analyses of greenhouse gas emissions over long term. And what we see is that there's a strong correlation between the rainfall gradients and the amount of emissions that are coming through. So essentially, New Zealand's soils are being developed over millennia and are being influenced by these rain gradients. And so again, if we look at a map of New Zealand, we see that there's a very strong gradient from west to east coast in terms of rainfall regimes. And these are directly correlated to different types of emissions or different ways in which emissions are generated in all of our soils. And it shows a strong correlation between the potential N2O emissions and the amount of rainfall that we're seeing. So very high rainfall areas tend to have lower emissions than lower rainfall ones. And it's probably because they've evolved different types of microbiome that lead to changes in the nitrogen cycle. But the thing is that we can't steal information from the rest of the world because our country is so different and our farming is so different that it's not really comparable. And it wasn't until a short time ago that we started really pinpointing the fact that we can't just learn from other people's projects. And so one of the take-home messages for today is that New Zealand soils are really unique. And so we need a New Zealand-specific model system to understand agriculture and the impact that it can have, both as a tool or as a source of emissions. And so to do New Zealand justice, we need to use a New Zealand-specific model system. And that's basically urine patches, those areas that are impacted by the source of nitrogen into our soils. And so all of you probably are well aware of urine patches. Each one of these represents a spot where a cow took a pee and basically loaded lots of nitrogen into that area. And we're creating a completely new ecosystem in those areas that break all the rules that we understood for nutrient cycling. And so what have we learned so far? Very fast, high-level overview of what we've learned. So the first thing that we need to understand is that urine patches are triggering a succession of changes in soils. It's reshaping the environment and creating completely different rules. And so the first thing that happens is what's unfortunately named the urine cascade, which is the series of transformations that happen over time within those urine patches. And so the first thing that tends to happen is that you have a liming effect. So this was an experiment where we were working with partners in Ireland, which is a very similar country in terms of its management of agriculture and its landscape. And so we took two soils and we either exposed them to urine or didn't expose them to urine. And then we also modified the moisture so that it was either high moisture or low moisture. And what we can see is that in urine-treated soils, you have a liming effect. pH goes up as urea gets hydrolyzed. And then as it gets converted, the pH drops back down. And that's correlated to the transformation of nitrogen from ammonia to nitrate, nitrite, and eventually over here to nitrous oxide. So it's showing you the transformation. But at each time point, we have a completely different ecosystem. And so that's going to end up selecting for different microbiomes. And so what we did was use DNA sequencing to track the microbiome changes inside of those urine patches. And the first thing that we found is that the number of organisms in those urine patches drastically decreases. So this is the diversity of microbes, and we essentially lose half of the organisms that are in those soils in terms of the microbiome. They're basically selected against, and you have a much simpler community. So you're losing some of your biodiversity. Now, that shift from a non-treated sample to a treated sample creates a completely different microbiome. So again, the dots represent the microbiomes. The closer they are, the more similar they are. And you can see here that you have your background samples, and then you have your urine-treated samples, and you can further subdivide them in high moisture versus low moisture. So we're seeing a completely different microbiome that doesn't look at all like our native soils. And the crazy thing about it is that even after 63 days, we see absolutely no recovery. And so you can imagine that in a field, you're actually going to be consistently exposing soils to urine. And so your community has completely shifted and may not recover. And we don't know the answer in terms of how long it would take it to go back to normal. So that's one of the big unknowns at the moment. But these were Irish soils, so completely different. We wanted to see if this still applies to New Zealand. So we repeated the experiment using New Zealand soils. And so in this case, what we ended up doing was having controls that had no urine applied to them. And then we had samples where urine was actually applied to those soils. The difference in this experiment was that we wanted to pinpoint what are the organisms that may be directly contributing to N2O emissions. And so to identify that, we blocked different parts of the microbiome. So we applied antibiotics. We either applied antibiotics that targeted bacteria or antibiotics that targeted fungi. And so we added antibacterials, antifungals, or a combination of both. And you can notice here that irrespective of the antibiotic that we put onto those soils, we actually still see urine as being the biggest disruptor of microbiomes instead of the antibiotic. So urine has a bigger effect on the microbiome than even killing portions of the microbiome, which is a bit surprising for us because it's not really what we were actually expecting. Now, if I actually remove all of the background controls, what we see is that under background conditions, you have microbiomes that are here, and as you impose different selective pressures, you can shift the microbiome. So it just is a case example of how the microbiome basically is giving you an idea of the selective pressures that are happening in any given ecosystem. And in this case, by applying antibiotics, we've shifted the selective pressure and we get different microbes. So we know that we can modify the microbiome by imposing some form of management. I'm not saying that we want to apply antibiotic to soils. It's just a case study to show that you can shift the microbiome. And the thing is that these impacts of urine are universal. It's not just the bacteria, it's not just the archaea, it's everything in that soil. So in this case, on the left-hand side, you have the profile for the bacteria in those communities, and the profile on the right shows the fungal communities in those exact same soils. And for both examples, what you're seeing is background samples separating from everything else that got urine treated. So again, it's showing that the way that we do agriculture is having a big impact on our microbiomes. And we don't really know or understand what the repercussions are for those. And so one of the things that we did this experiment for was to see if we can identify a key organism that we could target to actually minimize these emissions. And so by blocking different parts of the community, we can identify the ones that are more likely to give us a solution for the greenhouse gas problem. And so here we have N2O emissions measured under either urine conditions or background conditions. You see that in the presence of urine, you do see a spike in emissions at about 40 days. But if we actually inhibit the communities at that time point, either with antibacterial, antifungal, or a combination of both, we have a very strong decrease in the amount of emissions that we're seeing. So it's basically showing that the emissions are biologically mediated and that by blocking certain parts of the microbiome, we can reduce that. But it is extremely challenging because we couldn't find a single organism that was responsible for the emissions. It's a collection of organisms all responding independently that together lead to the emissions. And so in order to minimize the emissions, we can't just target one organism, you have to target the entire community. And so just to sort of wrap it up and give you a summary of what we've presented today. So basically we know that soil microbiomes are driven by habitat changes. So you change the habitat, you're going to change the microbiome. And we know that things like rain, soil type, land use, and even management can change the microbiome in soils. But for New Zealand, if we want to really understand something that's directly relevant to agriculture, we need to focus on these urine patches because they're the source of the emissions, and it's also a completely different ecosystem. And so this unique environment is going to change over time. It's not the same thing. Over time, as the urine cascade progresses, conditions change, leading to selection of different microbes. And basically, these changes in the ecosystem are reshaping the mechanisms and rules that control N2O emissions. So basically, New Zealand is unique, and because of that, we need unique solutions that are New Zealand-centric. We can't just rely on reading the literature in other countries because it just doesn't apply to us. And just to wrap things up, this was not done by myself. It was done by big collaborations with people from other universities or institutes here in New Zealand or internationally in Ireland or Norway. And most of the work in the lab is done by very talented graduate students and postgrads. And I just happen to stand in front of you and take the credit. But if you have questions, I'll try to answer them.